I want to thank the organizers for inviting me to talk tonight. Thank you guys for coming. It's an honor to be here and tell you guys the story of my trajectory and through my educational path that led me to this point, and then what we're studying now, and how we love to connect with you and the local community at VTCRI to foster some of the things that Natasha mentioned to help enrich some of the endeavors that you're pursuing in your class. So I'm an assistant professor. I've been here about 1.5 years, so I'm pretty new to the area, though Virginia holds a dear place in my heart. I'll let me back up because my wife, so I practice with my wife, so she told me to make sure I emphasize these themes, because she said these are clear throughout my presentation, so make sure I highlighted the curiosity, creativity, communication, and coping skills, we thought we'd keep it all C's here, that it takes to be a scientist and pursue science as a career. So along the path I learned many of these things, and so share a little bit with about that with you today. So like I said, Virginia holds a dear place in my heart, because I've grown up here. I grew up in Richmond, Virginia, and attended Manchester High School in Chesterfield County. So I wanted to share my path as well to maybe give you points to connect with me personally, because I feel like personal connections might help foster professional ones as well. So started in Manchester High School in the south side of Richmond, and then went on to UVA in Charlottesville to get a bachelor's in electrical engineering. But during that time, I realized I didn't really like electrical engineering. I actually thought I was going to do that and be smart and go take the pre-med courses and sneak into med school that way. Turns out I didn't really like the med school classes either. It wasn't as competitive as some of those folks are. But what did catch my interest was biomedical engineering. So I minored in that and went on and got a master's and a PhD in biomedical engineering at UVA. They let me stay there. And then after 10 years, they said, okay, go be somewhere else. And so I migrated not very far, but down south to UNC, Chappell Hill. Lived in Durham, so in case you're familiar with that area, it's sort of a tight-knit area, and there are lots of cross-collaborations and competitive rivalries down there as well. But lived in Durham, worked at Chappell Hill, and did a postdoctoral fellowship in developmental biology, so basic biology department. So yeah, I saw some eyebrows raised. It was definitely a shift from engineering to basic biology. First 3 or 4 months, I was hyperventilating in the bathroom, like, oh my gosh, what have I done? But my advisor was very kind and helped me learn a lot, and I've been very grateful. So I spent 7 years at UNC doing a postdoc. Traditionally, you should do a 2 or 3 year postdoc, but again, she was very kind mentor, let me say, for 7 years. And that helped bring me to my own faculty position here at VTCRI in Roanoke, and I also have an appointment in biomedical engineering in Blacksburg. So what's great now is I get to take all of this sort of varied background and blend it all together, take engineering principles and ask what can we learn from developmental biology that then we can integrate into an engineering perspective to think about developing therapeutic strategies to help human disease. So, nice little segue here to my slides that I call Why I Care About Blood Vessels and You Should Too. So blood vessels are obviously important throughout, been appreciated throughout history of scientists. Back in the days before they could even, actually take images, they were sketching what the vasculature looked like, and they appreciated the father of modern day anatomy, Andreas Vesalius. Is credited for basically associating the blood vessels, and this is taken from a review that shows vessels and nerves closely aligned. So that was appreciated even at this point how important blood vessels are for many tissues, but especially nervous system development. And there were a lot of developmental models developed to try to understand how the vasculature works, and that there's flow away from the organism and it has to be returned, and how the whole circulatory system works. There was a while at which it was thought that blood originated from the tissues in which the vessels were in. So it's, they had to discover that it's a circulatory system, and so we sort of stand on the shoulders of those who, kind of discovered those things using pretty clever models. So now we appreciate how important blood vessels are, not just for human development and tissue development, but how important they are in different disease states. So solid tumors are known to grow blood vessels by secreting high levels of growth factor. And so growth factor basically induces tumors to recruit blood vessels, and that's bad because it accelerates tumor growth. And it's also bad because it allows the tumor to metastasize to systemic locations. And that's bad for prognosis going forward. Blood vessels are also important in other diseases such as diabetic retinopathy. I was going to look up the fact, I think it's 52 people every day go blind because of diabetic retinopathy. And so this is a situation where blood vessels essentially grow uncontrolled over the part of your eye that receives the light. And so it's obviously a devastating illness, and blood vessels are the heart of this pathology. So folks in my lab and other folks we collaborate with are trying to understand some of the basic processes that drive this. And I'm also, by the way, I'm also more of a collaborative speaker, so if you have any questions during my presentation, please interrupt. So just want to put that out there. And one of the last situations that my lab is starting to look into is a situation where now that we can keep prematurely born children alive early and earlier, we're finding that there are some consequences of that, and one of which being they're higher risk for intracranial hemorrhage. So the vessels in their brain are still immature, still developing, so the trauma from actually being born and then trauma being in a new environment causes their vessels to be at higher risk for leaking and actually rupturing. And this is obviously detrimental for the child's overall survival and health, and can lead to behavioral and cognitive disorders later in life. So we're trying to understand how can we maybe rectify some of these early defects in the vasculature of the brain to then help the child go on to live a more healthy life. And so, going to go into the weeds a little bit. My wife was like, easy here. Don't get too deep in the weeds, you're going to lose a lot of people because not everybody's a cell biologist. So let me know if I lose you at any point, but try to keep this sort of surface level, so not get too deep into the minutiae of what I study every day. But some of the cells that compose the blood vessels are endothelial cells. And so we can look at these, and these are techniques developed by people much smarter than me, physicists and chemists, who develop things that we could use to label these cells and actually see them and visualize them. So we can actually visualize endothelial cells in the brain, and they're labeled here in green. And what actually captured my attention when I was in graduate school was another cell type that surrounds the blood vessels, known as a pericyte. Bless you. Not a parasite, those are bad, don't get those. A pericyte, which means surround, right? So pericytes actually go in and surround the blood vessels to help stabilize them. And what we're finding is, through other high-powered technologies, like Debbie Kelly, who spoke last year, she uses a lot of this electron microscopy kind of imaging to visualize cell structures. We understand that they wrap around the blood vessels, stabilize them, and also do a host of other things that are important for blood vessel health. So they capture my attention mainly because they're kind of an interesting cell that hasn't got much attention in vascular biology community, and it's becoming more and more important in a lot of different pathological situations as well. So basically, to wrap up sort of the summary of what my lab really sinks its teeth into is understand growth factors. So there's one particular growth factor known as vascular endothelial growth factor, affectionately known as Vegf. So Vegf is one of the most potent inducers of blood vessel formation that we know of. And it's secreted in high amounts, especially from tumors. And a gentleman, Drew Folkman, many years ago thought that if we blocked Vegf, we could block tumor growth. And everyone thought, oh, here's the silver bullet, we're going to cure cancer. And it worked initially, but obviously it turns out biology is more complicated than we ever really think it is. And it has helped in many cases. So Genentech, developed a drug called bevacizumab that blocks Vegf, and it's actually shown some good clinical results. But what we found is that tumors then become resistant to Vegf, anti-Vegf therapies, and then actually become more aggressive in terms of their growth. So unfortunately, it wasn't the silver bullet, and so this growth factor secretion story is much more complicated, and we have much more work to do. It's secreted in high levels in this diabetic retinopathy situation, and I was surprised actually to find this out, because the vessels are leaky, and my graduate student actually brought this to my attention, because I was like, oh, because there's not enough Vegf, so the vessels aren't forming. She's like, no, there's actually more Vegf. So there's so much Vegf being made in the brain, because Vegf is secreted when tissues don't have enough oxygen. So the brain is just telling the body, give me more vessels, give me more vessels, give me more vessels. But there's so much Vegf now that it's actually having the opposite effect of what's developmentally supposed to be happening. And so we're trying to figure out how developmentally and these different pathological situations, how disrupted growth factor signaling causes blood vessels to form in a way that we don't want them to. And how can we target it in a more sophisticated way to treat some of these diseases and hopefully provide some measure of a cure. Okay, so hopefully that was sort of an overview of like the main bits of my science. I didn't see too many glossed over faces, so I think maybe success. I'll tell my wife the good coaching she gave me. But what I did want to do is also kind of step through, and I know being a teacher is sort of a thankless job. And so I'm going to get choked up. Every time I've talked through this slide, I get a little choked up. Remembering the folks who have kind of guided me through my career, Mr. Henry, as a chemistry teacher in high school, and inspired me to take on chemistry as a junior, and then tricked me to take AP chemistry. And man, that was a lot harder. Didn't do as well in AP chemistry, although he did propel us both with the AP test, I guess. So that was great. Grateful for him for that. But he was such a funny guy, but he definitely inspired us, all of us, to love chemistry more than I think we initially thought we would. The turning point for me from going from electrical engineering to biomedical engineering was certainly Dr. Bill Guilford. So he's a cell biologist who studies molecular motors. And he showed us his video, and that's why I want to show a lot of videos today. Because basically it was things that I'm like, just have my mind blown as an undergraduate. Basically he showed these fluorescent proteins in a dish, and they're just sitting there, not moving. And he adds a little bit of calcium, and all these molecular motors just start moving down these microtubule chains. I'm like, you're telling me you can get motion just by combining molecules together with some ions? This is like, what is happening? And it just blew my mind. I was like, I want to study this. I want to understand this. And then I thought, well, if I can understand, help study this, I actually can help human health. That's why I wanted to go to med school to begin with, and by hopefully discovering ways that we can design better drugs and cures for people. So that many thanks to Dr. Guilford, who's still at UVA, I think teaching and doing some pretty awesome science as well. And then, as I mentioned, this very generous woman, Dr. Victoria Bouch, who let me come to her lab as an engineer and learn a lot about developmental biology, make a lot of mistakes, and teach me about just developmental models and biology in general, as well as let me stay for 7 years. So she spent a lot of grant money on me, but hopefully it was worth the investment. And she's helped me, in my understanding of developmental biology, which is now sort of the foundation of my lab going forward. Okay, everything seems to be playing a little bit earlier, so I lost the wow factor here. But so what I study in my lab are developmental models, and one of the developmental models we use are mouse embryonic stem cells. And so we can differentiate these stem cells, and they develop into different kinds of cell types in the body. So that was first step one. Oh my gosh, what are you talking about? They'll literally develop every cell type. And second, a lab technician came over to me one day. She's like, come here, and I started in the lab. She's like, look at this. It's amazing every time I see this. And I was like, what do you mean? She's like, look harder. And I'm looking at it, and I'm like, are those cells? Is that really a heart trying to form in a dish with just cells? She's like, yes, it is literally a heart trying to form. It's not in an animal. It's just differentiating stem cells in a dish. They're linking up electrically and starting to conduct signal together. They won't eventually form a heart because they're not in the right context for that. But it's literally trying to form a heart. I was like, what is happening? Again, as an engineer, my mind was just blown. Like, I didn't realize this sort of thing happened in a dish.
So is that just with a monolayer of cells, or is it how big is that?
So they're funny. It's a good question. They are actually cells that we form. We make them form a sphere. And then we set that sphere down on a plate, and it spreads out. Now, if you took that sphere, I don't know if actually you took that sphere and embedded it in the animal, if it actually go on to form an embryo, I think it actually would. I'm not sure. We haven't done it. But
PC12s?
What's that?
Are they PC12 cells?
They are. So I don't. Again, engineer. R1 is what they're called. Told me.
Yeah, sounds different.
Sounds different. I don't really know. Again, engineer. They were like, here, use these cells. I was like, got it. So this was me being curious and wondering and being amazed. But there are a lot of technical things that I didn't know, just like put growth media on it. Yeah, this happened. So it's more than a monolayer, though. To answer your question, it's as the cells spread out, it's several layers of cells on top of one another. So one of the other cell types that develops, and I told you I'm more of a vascular hound when it comes to science, are endothelial cells. And so we use, again, some techniques and tools that were developed that were outside of my expertise, but other people developed them and did genetic tools to actually visualize the endothelial cells as they're forming blood vessels. So these are differentiating mouse embryonic stem cells forming vessels, and we can watch as an endothelial cell is reaching across here, these very thin processes called filopodia, reaching across, grabbing onto the vessel, and we'll see a cell divide here. There it goes, into the two nuclei. And so I'm watching this happen, and I'm like, okay, first of all, the heart's beating. Second of all, endothelial cells, I mean, this is without blood flow. So there are inherent genetic mechanisms in these cells that are driving it toward making a blood vessel. It wants to develop a living animal, regardless of whether there are other, blood is actually flowing. Because this is not with, again, like anything in tech anymore, blood is flowing. It's just happening by itself. So, okay, this is, again, one of those moments of like, wow, this is a moment of discovery for me. And then can we describe it in terms of, what the biology is occurring to help advance science? So one of the other movies that we look at are how blood vessels that are sort of in single file, endothelial cells kind of all lined up, they eventually have to acquire the space for blood to flow. And when I saw this movie, it blew my mind for a couple of reasons. One of which, and I don't know if you noticed what happens with all these vessels, almost at the same time. Does anybody see what happens? They're connecting, they're fusing, that's right, yeah. And they're opening up at the same time. So when I was in grad school, they taught me, it's sort of a zipper effect. You're along a line of cells, it should just move downstream. This is actually happening all at the same time. But we don't actually know why that happens. My postdoc finally ran out. My wife's like, go get a job. This is so cool. So I don't know if anyone's going to figure this out. There are a couple people who have proposed ideas in Europe, but I don't know if this is exactly what we're seeing here. But it blew my mind to see all this happening in different spots at the exact same time. Okay, so this was another phase of blood vessel formation. They have to sprout, they have to open up to allow blood vessels to form. Okay, and then the last bit, this is where my true love now comes, is pericytes. The lovely pericytes, here marked in red. And they are attached to the endothelial cells in green. Now we're moving to a little bit of a different model. This is where we use mouse embryonic tissue that we've explanted and embedded in a matrix. And we still use live imaging techniques to keep the tissue alive, but they actually just let it continue to grow and develop. And so we can actually watch as the vessels, after they kind of settle down, they're like, okay, we'll just keep doing what we were doing. And so they start to sprout out. Sorry, the lighting's a little tricky in here because of some of these aren't showing quite as well. But what you can see in some of these areas is the endothelial cells reach out and start to look for places to connect. And the pericytes are like, oh yeah, let's do that. They're like, whoop, reach out, and then that kind of didn't go anywhere, so the pericyte's like, all right, we'll back down. And then, let's see, what is this one? There's a pericyte that's, oh, thank you, there you go. And there's a pericyte up here, and it's like, okay, this vessel's getting longer, it needs some friends. So it continues to kind of march on down the line. And a lot of this, how this happens, is not really well understood. How pericytes and endothelial cells can talk to one another, how they migrate with one another, is still kind of being worked out. And it's a very important process, like I said, in a number of different diseases. So we're very urgent to try to figure some of this out so we can potentially develop drugs and therapies to help with those diseases. Okay, now lights back on. Okay. I'm going to take a little bit of a risk here. I told my wife, she was like, no, you shouldn't do it, it's cheesy. But I saw some of you already tired. I know you're tired, it's been a long day. If you'll indulge me a little bit of active learning. Is that cheesy? No? Let's do it. All right. Thank you. All right. I appreciate it. All right. Well, then whoever said let's do it, we need 6 volunteers. So let's do it. Let's do it. Why don't you guys come on up here? Yeah. Come on. Stretch the legs. I need a couple more. I need 3, 4. I need 2 more. Just to get your blood moving, because you need blood to move. That's helpful. All right. You're differentiating right now. Congratulations. You're an endothelial cell. You get to be up there. So. You don't have to know anything. You don't really need to know anything. There we go.
That's good. I qualify.
Yep. Endothelial cell.
One more.
Congratulations. Appreciate it. All right. Excellent. Wow. I really appreciate you guys humoring me in this. Okay. Now, you're almost even halfway there. But what I would challenge you is endothelial cells have to form a line, and pericytes have to provide a little bit of current coverage. So, ready, go. Endothelial cells. Okay, there you go. Wow, you're faster than development. My experience is fast. I would be a genius.
What about the other side, though?
Oh, exactly. Well, the pericytes.
Do we have to wrap around?
I don't know. There's a pericyte decision to make. Does a whole pericyte move around to the other side? You can just walk around, maybe. Maybe that's sufficient. There you go. There you go. Perfect. Blood vessel. Exactly. Let's get the volunteers. I appreciate that. Sorry. So why I wanted to do that, I do that like at science festivals and sometimes with undergraduates and graduate students to get them up moving around, thinking about what's going on. But what had to happen for that process to occur? Communication. Exactly. So that's one of the things we try to drive home with students is that cells have to communicate. But it's not just simple communication like that we can do, right? But even human communication involves what?
Recognition.
Words, recognition, language, all these different elements. And it's just as complicated for cells, right? They have to negotiate their environment, they have to negotiate each other, they have to ask, if there are two pericytes on one side, is that how things should be? We don't know. Cells might have that figured out, because when I look at my movies, those pericytes are only at branch points. How do they know to be only at branch points? We don't really know, right? And so those are the kinds of questions we ask students to inspire them to say, we need your help to figure this out. The scientists, smarter than we might sound sometimes, or we think we sound, and try to create a little job security by creating big terms to make us sound smart. The reality is, it's just communication. It's just cells moving around trying to figure out how to be where they want to be, so that the tissue continues to form as it should. And then when communication breaks down, you get diseases. Cancer. A cell goes rogue, essentially. Others, if a cancer cell starts to become cancerous, a lot of times the cell neighbor will go, hey, you're not where you're supposed to be, punch, you're dead. And that happens all the time, actually. You get cancer all the time, which other cells tell that cell to die. And so when that one cell stops listening to its neighbors, that it becomes a problem. So I try to drive that point home with, oops, set down my pointer. Is that it? Oh. Because I got the same pointer in my pocket. There we go. Okay. Sorry. So we try to drive that point home using, trying to use different analogies. And so I appreciate you guys indulging me in that way. It might be something that you could take back to your class if you're in the biological sciences, to kind of demonstrate to your students. I was actually thinking about it, I'm like, actually, I don't know if high school students would be amenable to that, because I know they're sort of bundles of energy, and you probably don't want them to be too close to each other, because they're going to freak out, right? But maybe you could take it back to your class and use it as a tool to set up, some of those movies. And I'll get to that sort of connection in a bit. But when I received the invitation to speak tonight, there's a long description of things to talk about, one of which was what's needed for future science candidates. And so I just sat and thought about that. And the reality is, you're already doing the basics, right? You're already teaching science. The basics of science, physics, chemistry, math. You're already doing that. So I'm not going to tell you that, because you're working hard every day to do that. So what are the other things that would be good to try to convey? And I know it's so hard. I have a first grader now who's already learning to take the test, right? Kids now just learn to take the test. And gosh, does that just kill like imagination and creativity? If not, it's just such a disservice to inspiring the next generation of kids and scientists and learners across disciplines. But if you can, and I know it's so hard, but these are the kinds of things I would say. In addition to the basics, curiosity and wonder. Just innate human curiosity to ask why. Why does that happen? Why is the wind blowing? Why is it so cold? Why are those cells moving around? Why? I think that's one of the big things. When I sat down and looked at in addition, like, why are those cells contracting together? I don't really understand. Then I went and looked, and I looked up on Wikipedia. Electrical conductivity among cardiomyocytes. Like, okay, great. So people have figured this out. But then I look up about pericytes. Why are there only cells at branch points? Well, it turns out Wikipedia doesn't have a lot to say about that. They have some ideas, and even like the primary literature doesn't really know. They have some ideas, but we don't quite know the full story. So then the other part of that is creativity. And as I was thinking about this, I was thinking, the creativity does not just ask why, but then take it to the next step and say, propose how you think it might work. You might be wrong, make a mistake. That's okay. We do that all the time. I'll get to the last one on that. But that's a hypothesis, right? Why does that work the way it does? You tell me why. Why do you think? My son and I do this all the time. And usually, he comes up with this crazy idea that I'm like, that's actually kind of reasonable. I'm like, you're only in first grade. That's actually kind of like in the realm of what could happen. And then the ability to explain it to others, and that's where my kooky idea is that we, as educators, and I'm teaching, undergraduates and graduates, a lot of times it's if you told them in sort of the rote biological terms, it's sort of like gloss over, I'll write it down, but I don't really internalize it. But if you give them an analogy for them to kind of wrap their mind around, I do this with my graduate students all the time, we anthropomorphize everything. We make cells like humans. Why did that cell decide to do that? Well, it didn't decide, but that's sort of the language we can use to help them understand like what a cell is sort of after. And then, communication skills. And I know this isn't, probably in your wheelhouse in terms of, science and math and engineering kind of teaching, but it is so important. I do so much writing now, so much writing. I actually am not at the bench much. I'm really actually doing a lot of writing because I have to write up what we've seen and then submit proposals, the hypothesis. So writing becomes very important to articulate and formulate a hypothesis that other people can understand. And then public speaking. I took extemporaneous speaking when I was in high school, and that was good, even though there was a moment, I definitely had a competition where I just quit. I just left the room, and my coach was like, why did you leave? I was like, I was nervous. She was like, don't do that again. I was like, okay. So she made me go back to the next round after I was not going to win. And I did it, and I appreciate it. I'm so glad she told me, encouraged me to go do it. Because I think that was, served me well in terms of being able to speak publicly. So then coping. Coping. Boy, do we fail a lot. We fail all the time. And I see that in my son. He hates to be wrong. He hates to venture a guess because he doesn't want to be wrong. I'm sure you guys face this in your classes when you ask the whole class, what do you think about something? Silence. And I'm sure that's just, probably irritating. But scientists fail all the time, right? And I told this to my wife. She's like, you should tell everybody that. When I started electrical engineering, my first exam, I got a 26. 26 out of 100 points. 26. I should have quit right then. I should have gotten the message. But the average was a 32, so I was like, okay, we all still get this. But that was pretty discouraging to get a 26. I left high school thinking I'm pretty good stuff, and I got to college thinking, whoa, boy, this is hard. So science, it requires the ability to deal with failure and learn how to cope. And I think that helps in life as well. You're going to fail sometimes, but just giving it a try. Okay, so then ways to connect. I think Natasha already mentioned some things that, we're all about. We want to be integrated in the classroom as much as we can in every way we can. I know not everyone here teaches biology, even though that's sort of my discipline. I think there are ways that we can still figure out connection points. If you do teach biology, or there are ways that we can connect via chemistry or whatever it is that you're trying to teach in the classroom. We'd love to visit your classroom, or something that I set up with my sister-in-law, actually. She teaches 5th grade in Williamsburg. And we do something that, sort of, I guess we kind of just were chatting about it one Thanksgiving. And she was like, would you be willing to do this? I want to try to help get my students inspired about science. And she said, would you, this thing's a little trigger happy, there we go. Where she gets all my videos, and she shows them to her class, and she just introduces cells. Like, what's a cell? What are the different parts of a cell? All those sorts of things. And then she shows them my videos and says, these are cells in the act of making a tissue, or making an organ. And then she gets them to kind of talk about it, think about it, and then she says, now we're going to interview the scientists who created these videos. So she got the whole 5th grade this year to file into the library, and we connected via Skype. And so that was much easier for us to visit her class. There's no permission slips involved, nothing like that. We just set up a Skype. It's super cheap. And myself and Dr. Sarah McDonald at VTCRI just shared our experiences, and they asked us all sorts of questions I didn't even know the answers to, which was fantastic. It was so great, because it was like, yes, this is what we want. We want to inspire students to ask questions. And I think this was a success, and now we're going to be making this an annual thing. And then after this, I basically picked up her iPad and walked over to my lab and showed her showed the whole 5th grade different parts of my lab. Microscopes, the cell culture room, which is great because, I couldn't bring a whole class in there because of, regulatory issues. But I can take my iPad in there and just kind of show things off and, show my lab members doing stuff. So that gave students the context for this is what science looks And these are the kind of cool microscopes we get to use and things like that. She said a lot of students really responded well to it. Except this guy, he wasn't paying attention at all. No. So in VTCRI, we have some other things that we'd love to continue to expand. We have the VTCRI Brain School. We tried to do the Heart School last year. It snowed, and then I think we lost a little momentum on that. But hopefully next year we'll get some more organization around that. We do our summer science camps. I'm not involved in those just yet, but we're continuing to develop those. So hopefully, now that I know where things are a bit more, being a new guy, we'll I get more involved in these things. Like I said, we want to share materials and resources. If you have microscopes, if you need software or something like that, we'd be happy to try to figure out solutions like that because we can have academic licenses that we can maybe tap into. Mentoring, internships, and volunteering, absolutely, absolutely. If you have some bright students who either like biology or sort of anything, we could probably figure out a project for them to work on. It's good experience for the students, obviously, and then from our perspective, it is great for grant submissions. Even right now, and I'll put this out as a public call, I'm submitting an Nsf Career Award. And what they require for that is not just a research component, but also an education component. And so I'm designing an educational component for an undergraduate course, but also to have an outreach component to that. Nsf loves that and would fund that and provide money for that. So that would set up the infrastructure for this conversation between Virginia Tech, VTCRI, and local schools to actually get some money behind it, some real teeth to it. So absolutely, we are here to help in any way that we can. Because the other thing I'd say, within that NSF award, that's the detail I was going to mention, I'm doing computational modeling. So if any of you have some sort of engineering, computational elements to your classrooms, curriculum, I would definitely appreciate a high schooler, because some of the software we use is actually designed for high schoolers. It's called NetLogo. And it's literally easy to use. It's like so easy to use.
Yeah.
And you can actually do some really powerful things with modeling how cells interact with one another. And then we integrate the molecular layer through some other modeling approaches, and it becomes a very powerful tool to make predictions. So, just a big plug for that from my lab. Okay, so then, last but not least are the people who helped me do some of this science, and these are all the folks in my lab as we're kind of growing. Definitely mention Sarah Taylor. She came with me from UNC and has been vital for getting the lab off the ground, so I thank her pretty much the rest of my life. She's done so much. And then my graduate students who indulge my corny analogies and whatnot, and the other folks in my lab. But with that, I'll take any questions now, but I guess we have dinner coming up and opportunities to connect then. So, and I brought tons of cards that I rarely get to pass out, so hand them to all of you. But thanks so much for your time.